Now that we've discussed all of Mendel's rules of heredity and the exceptions to his rules, such as codominance and incomplete dominance, we can look at blood type and sex-linked inheritance. We'll start with blood type. And blood type has multiple alleles, and this happens in a couple of the different traits, such as skin color in humans. And there's 4 different blood types that we could have: A, B, AB, or O. We call them the ABO blood groups. And it actually has to do with the antigen or the protein that's present on the red blood cells. So you would either have the A proteins, the B proteins, or no proteins, no antigens at all on your blood cells. And to show this, we have these different alleles. There's a couple of possible genotypes. Here we represent the alleles with this capital letter I, and then the type of protein that we're talking about. So here we could have homozygous type A or heterozygous A, which actually means that you carry the allele for O, which is represented by this little I. B is the same thing. We could be homozygous for B or heterozygous and have the O allele. AB has only one option. You have both the A allele and the B allele. So you have both proteins on your red blood cells. Or you can be type O, which means that you have no antigens on your red blood cells and you're represented by these 2 little I's. Now, to determine blood type, it's a very easy test, and it's just an antigen-antibody reaction, because in your blood, you have the antigens on the red blood cells. Those are either A, B, or none. In the plasma, you actually have antibodies for the opposite type. So if you have the A antigen on your red blood cell, you have type A blood. But in the plasma, you actually have antibody B. So what happens is if you're given the wrong blood type, agglutination occurs, and that is fatal because the blood can't move through the blood vessels. And agglutination just refers to clumping. And if you look at this test here, we can see how these are nice and thin, and then this gets all clumpy. Imagine that going through your blood vessels. And if you needed a transfusion, we'd want to make sure that that was proper. And a transfusion is just getting blood from an outside source. If you are type A blood, you can receive a transfusion from somebody that has type A or type O, preferably A. And if you were type B, you could receive a transfusion from somebody with type B or O. Type AB is actually called the universal recipient because they can receive blood from any type. And the universal donor is type O. They can give to any type of blood, but they can only receive O. And there's one more antigen that might be present on the red blood cell, and that's the Rh antigen. That's what we actually represent in this third well. And if you're positive for Rh, you would have type A positive blood if you're type A, and if you did not have it and you were type A blood, you would be A negative. And to tell, what we do actually is we put the blood in these 3 wells, and you can see there's a well for A, a well for B, and a well for the Rh. And then we add serums to them that have the antibodies in there that would react with type A or B or Rh, because it's going to be an antibody-antigen reaction. So we would put an antibody that would connect to the antigen on the red blood cell for A. So we would put the A antibody in. And if it clumps or agglutinates, we know that that's the blood type that it is. So this actually shows it's positive for A. And then we would do the same thing for B. We would put the blood in here and then put the antibody for B in the well, and we also get some agglutination. And then we would do the same thing for Rh, and we have agglutination. So this person's blood type actually would be AB positive. Now, let's do a test cross with blood types because this can get a little confusing. We'll say one parent is heterozygous for type A. One parent is heterozygous for type B. Possible outcomes could be AB, type B, A, or O. So our phenotypes are 25% AB, 25% B, 25% A, and 25% O. So it's possible that these parents could have a child that has a completely different blood type than they do. Now let's look at sex-linked inheritance. And first we need to look at what a pedigree is in order to determine this. And a pedigree is a chart that shows the generational flow of a family. And we start with the first generation up top, and all these shapes represent things. So a square represents a male, and circles represent females. If it's filled in or colored, that means that the person is infected with whatever trait that we're looking at. And if it's half filled in, sometimes that's going to refer to a carrier, which means that they actually have the allele that can be passed down, but they don't express it. Let's look at hemophilia. This is an X-linked trait, and hemophilia, remember, is a disorder where blood doesn't clot. And let's just refresh our memories that if we have an XY chromosome, the person is a male, and if it's an XX, it's female. Now with X-linked inheritance, it's kind of interesting because if the um, allele that causes the disease is on the X chromosome for a male, then the male has it. If it's on an X chromosome for a female, but the other chromosome has the normal allele, they're just a carrier and they don't express the actual disease. So here we could see this X and the little h is going to represent hemophilia, and Y, this male is a hemophiliac, but this male is normal. This female here with the capital H and the little h is a carrier, and if she had two little h's on her X chromosomes, then she'd be a hemophiliac. So let's look at a parent that is a carrier, and the father will be normal. Our outcome, we would have um, a daughter that would be normal, another daughter that would be a carrier, a male that would be normal, and a male that would have hemophilia. So the chances of having hemophilia in this case are 25%, but there's also a 75% chance that the child born to these parents would not have hemophilia. So when you're looking at X-linked chromosomes, you have to remember that if it's present with the male, because they only have one X chromosome, it's there, and they're going to express it. But if they have one bad allele and one good allele, then they're going to be a carrier in the case of this female here. But you do the cross to see if it's possible for the offspring to have the disease.